Namaskar. Myself, Dr. Sindhu Gm, Assistant Professor of PG Department of Botany, JSSCACS, Mysore. Today, we are going to discuss about the topic seed production techniques. This topic comes under paper title Hardcore 2.3 Plant Breeding and Evolutionary Biology. So, in this topic, we'll be able to cover two course outcomes, and here we'll be learning the in-depth about plant breeding methods and techniques. Also, course outcome 2, understanding the details of breeding for specific purposes. Before going to the topic, there are some basic aspects which we need to glance upon before diving into the actual topic. First, we'll be discussing what is a seed. Seed, it is defined as a mature integument megasporangium or mature ovule, which consists of embryonic plants, stored food material, and it is covered by a protective coat. This is called as a complete seed. And this seed are of different sizes, different colors, depending on the plant, depending on the variety, it differs in its structure. So the major four goals of seed technology are rapid multiplication, timely supply, assured high quality of seed, and reasonable price. Rapid multiplication is the major means in plants' asexual reproduction. In sexual reproduction, seeds are produced, hundreds and more than 100 seeds are produced per plant, which each of the seed has the ability to give rise to a new plant, and it has the totipotency to develop into a new complete plant with certain environmental factors. And seed technology also plays a major role in rapid multiplication and providing the seed materials required by the farming community or by the market on time. Timely supply is the second goal, wherein the required variety and in required quantity, the seeds will be supplied to the farmers, to the market on time. Assured high quality of seeds. So already we have abundance of seeds produced by plant without human intervention. But with the aid of seed technology, we can assure the quality of seed to be optimized to its maximum level. Reasonable price is the fourth goal of seed technology, which mainly helps the farming community and the consumers with to obtain high quality seeds in lowest possible cost. So moving on to the criteria of quality seeds. How do we decide seeds are of good quality or bad quality? This is assessed by major steps, like it should meet minimum genetic purity. Genetically, the seeds has to be pure and it should perform well in the agricultural lands. It should have good germination. So seeds, whichever you use for plantation purpose, they have to have better germination, and all the seeds must germinate in the field. It should be free from infection or seed-borne diseases or storage pests. This is the major constraint or problem in the seed technology, where storage of seeds has become a major problem. And in this regard, seed technology is helping us to minimize the infection or to eliminate the pest and pathogen present in the seeds. And it should not contain impurities like other crop seeds, trash material beyond the permissible limit. Apart from the optimum levels, it should not exceed the permissible level of impurities. So with all these criteria satisfied, we call it as a good quality seed. Here in this picture, you can see that physical purity should be at least 98% in any variety of the seed. Genetical purity should be 98 to 100%. Germination should be as maximum as possible. Similarly, they have to be uniform in shape, viable in nature, and they have to be healthy. And these are all the criteria which decides a seed as a good quality seed. And these are 8 steps of seed technology, seed production techniques, where it starts from seed dormancy breaking. Seeds are dormant materials which do not germinate on its own. There should be some external force that trigger the germination of the seed. And second one is seed multiplication, seed processing or treatment, which includes seed drying and cleaning of the seed. Seed treatment, which includes disinfection or giving protection to the seeds. Fifth one is seed storage. Sixth one, seed packaging and handling process, and then seed certification, and finally seed marketing. These are the 8 mandate steps in seed production techniques which has to be fulfilled for a variety to reach the market and to reach the consumers. First, we'll be talking about seed dormancy. All the seeds have common structured organelles in it. For example, the seed coat, the internal aileron layers, endosperm, embryo, and some storage material in it. So these are common parts of the seed. So dormancy is defined as non-germination of the seeds due to absence of suitable conditions is termed as dormancy. Or it can also be defined as a physical or physiological conditions of viable seed which prevents germination even in the presence of favorable conditions. Dormancy naturally is a favorable process where it helps in long-term storage of seeds. So seeds stay ungerminated as long as the favorable environment is not achieved. But dormancy is sometimes harmful in terms of when we are requiring the seeds to immediately germinate and grow into a new plant. Then dormancy has to be broken with other agencies. So what are these processes or methods that are used to break seed dormancy? First method is scarification. In scarification, there are three types, where in the first type, the seeds are either rubbed on a sandpaper manually. So we rub the seeds and break the seed coat in case of hard coated seeds, where water is not entering into the seed coat, wherein permitting the further process of mobilization of nutrients and replication process being initiated. And the second type is the seed coat has to be removed completely by clipping or cutting the outer seed coat. We open up the seed coat for easy moisturing of the seed. And third one is soaking treatment, where concentrated solutions or diluted solutions of acids like sulfuric acid has to be used, where the seeds are soaked for 1 to 60 minutes to soften the seed coat. So then the seeds can be used for germination easily. Second one is treatment with growth regulators and other chemicals. Growth regulators, particularly gibberellins, cytokinins, ethylene, all these are certain growth regulators which helps in breaking the dormancy. Sometimes there are inhibitors present inside the seed coat which is inhibiting the dormancy, the germination process. And hence, treating with these growth regulators will remove those inhibitors and will trigger the germination process. And chemicals like potassium nitrate, thiourea can also be used for seed soaking and further breaking the dormancy. So this is the seed germination initiation process. In this, other two methods which includes temperature treatments as well as light treatment. Temperature treatments wherein the dormancy is because of embryo factor. The previous treatment is only for the seeds which the seed coat is harder. But this treatment is in case of embryonic inhibition for germination. In these cases, temperatures of low or high temperatures for certain period of time will be essential for breaking the dormancy. So, for example, temperature of 0 to 5 degrees Celsius for 3 to 10 days will initiate the germination process in certain seeds. In this picture, table, you can see that different plants have different temperature ranges, minimum, maximum, and optimum temperatures. So these temperature has to be met so that the seeds will germinate. Some seeds require brief period of 40 to 50 degrees Celsius before germination, and a few hours of treatment will be necessary. Here you can see few examples of beans which has minimum of 60 degrees and maximum of 85 degrees Celsius. The optimum temperature required for the bean plants to germinate is 75 to 85 degrees Celsius. Similarly, we have another example of eggplant which requires 60 to 95 minimum and maximum ranges, and the optimum level will be 60 to 85 temperature requirement for initiating the germination. And there is also hot water treatment where seeds are soaked at 80 degrees Celsius or above and for 1 to 5 minutes. This is very short term incubation which soften the seed coat or which will trigger internal processes allowing the seed to germinate. Last one is light treatments. Light treatments where some seeds do not germinate in dark conditions, where it requires continuous or periodic exposure to the light is essential. In this picture, you can see that there is a light source above the seed sample, and the seed sample is kept inside a glass chamber, a transparent glass lid, which has 98% humidity maintained inside, and the seeds are spread in a saturated potassium sulfate solution, which initiate the germination process effectively. Coming to the second step, seed multiplication. There are different stages of seed multiplication. Initially, when a breeder develops one new variety of seed or good quality of seed which is not existing in the market, such seeds are called as nucleus seed. It is an initial amount of pure seed of an improved variety which is available with the plant breeder. Usually, the tag of certification for nucleus seed represented with white color certification tag. Breeder seed, it is the seed obtained from progeny of nucleus seed, and similarly it has yellow colored tag. Foundation seed is an seed which is produced for seed multiplication farms like state and central universities, agricultural, governmental organizations who will be multiplying the seed for production, maximum multiplication of that. So this seed is again with white tag, and registered seed is which is the growers are selected from the progressive farmers, and the seeds are given to them to multiply it in large quantities, and that again is called as registered seed. And finally, certified seed, it is the progeny of registered seed which is ready to get certified and reach the market. So these are the different multiplication stages of seeds. Third method is the processing of seed, in which we'll be studying about drying methods. So we have wagon drying and crop dryers and various other dryers which can be used to dry the seed. And wagon drying, it is a wagon, a special type of apparatus which helps in drying the seed in large quantities. Bulk seeds are dried in batches where it uses heated air which is circulated throughout the wagon, which permits effective drying of the seed. Crop dryers, as you can see in the picture, it has a temperature regulator and there is continuous hot heat circulated throughout the seed samples. Seed samples are spread in batches and layers, which finally helps in effective drying of the seed samples. And the next one is the bag drying. So gummy bags are usually used in agricultural lands to package the seeds soon after processing, with removing after minimum impurity level is maintained. And these bags can directly be used to drying the seed. And typical design criteria which provides 25 to 40 cubic meters of air per minute is circulated in these bags. So these bags are effectively used for drying the seed, and it takes longer time than bag and wagon dryer. And also these dryers can be used only when there is small quantity of seeds or many varieties of seeds at a time has to be dried. Box drying is another method of drying where the box dryer, small box with perforated ventilating holes are present, as you can see in this picture. And these are plastic or glass bags usually used for this. They have many holes and vents, and using this locally available materials are fitted with perforated metal or woven wire bottoms. They are used to dry the seeds. Next step is cleaning seed. And in this, various methods of cleaning, pre-cleaning, pre-conditioning, cleaning and upgrading, all these are 3 steps present in this cleaning of the seed. As you can see that pre-conditioning is a step. Before cleaning, we have to prepare the seed. So seeds are prepared in such a way that they are coated with pre-cleaning agents to facilitate effective cleaning of the seeds. And sometimes the perforated meshes or sievers are used, as you can see in the picture. Different size and shapes of sievers are used to separate the seeds according to size and shape. And then after pre-conditioning, basic cleaning operations are done, as you see in this picture, wet processing and dry processing. So all these are some processes of cleaning of the seed, which I'm not going to study in detail. Upgrading the quality of clean seed. After cleaning also, we should ensure that seeds are still viable and have not undergone any stress, so that it will fail to germinate or it may lose its viability. Next is seed treatment. Seed treatment is done in different stages. First with seed coating, especially the soil in agricultural land contain a lot of pathogens or the debris from previous season or from debris of the infected plant. All these has to be eliminated from the seed sample. and especially Pythium species, Rhizoctenia, Sclerotium. All these are some pathogens that are present constantly in the soil sample. They may rot the seed before germination, or even after germination, they may kill the seed or seedling emergence. So for this, we'll be coating the seed with the ingredients with binder liquid, filler powder, and all these active ingredients are added to the seed coating. Our seed coating mechanism can also be done with 3 types of equipments: a fluidized bed, rotary coater, and rotating pan. So these coat the seeds in different formation and ranges. Other measures like sanitation and fumigation can also be very effective for seed treatment. Sanitation like DDT and malathion are mixed with a legally recommended dosage and sprayed, mixing with the water evenly on the seed sample or in the form of powdered sample. Fumigation, where gas tight storages are used, and the loss sometimes if the dosage is high or low, you can see the recommended dosage of methyl bromide in this table. Only 24 hours exposure at 16 to 30 O's per 1000 of it is recommended. If it exceeds the recommended level, even the hydrogen cyanide and hydrogen phosphide, it may harm the seed's germination. Even the seed may lose its germination or vigor. These are some equipments that are used for seed treatment. As you can see, the slurry treaters, direct treaters, homemade drum mixers, grain auger, shovel. So all these are equipments that are used in treating the seed. So these equipments, depending on the scale of seed treatment, large scale or small scale, different equipments will be applied. So all equipments are equally effective. Some are used for large scale seed treatment, some are used for small scale seed treatments. Next is storage of seed. So purpose of seed storage is to maintain the seed in good physical as well as physiological conditions where they are harvested and until they are replanted. Until then, seeds are effectively stored. The stages in this are storage on plants. Some seeds are stored on plants wherein they can be kept longer than after harvesting period. As long as possible, it is kept on the plant itself. Harvest until the process and store in a warehouse. This is a short-term storage, and in storages, proper warehouses are built for seed storage. Some seeds are stored in -degree temperatures, and some are stored in warm temperatures, dry environment. Depending on the type of seed, different storage purposes will be selected. In transit, for example, rail wagon trucks, when we want to transport the seed into different regions, will be storing in this transit, which is again a short term. Depending on the distance, it will be stored for the number of days. In retail stores, where until the consumer purchases the seed, until it reaches the proper destination, the seeds are stored temporarily in this region. On the user's farm, again, as the next season is available, the farmer is ready to store them. The seeds are stored in user farm. And here in this first picture, you can see the seeds are treated with hot water, and in the second, you can see the glass jar, airtight glass jar are used to store the seed. And at last picture table, you can see the crops and maximum moisture content you have to maintain to store the crop. For example, wheat, which requires 12% moisture content. Less than 12 may harm the seed. It may lose its viability and germination. If it is above 12, then it may also have become viable for infection with pathogens like pests and pathogens. Similarly, oats have 13%, barley 13%, sorghum and other seeds, percentage of moisture content is mentioned in the picture. Next is packaging and handling. So packaging and handling of the seed is again very important process. After following all these very sensitive precautionary measures, if we do not package them properly, everything will be of waste. The packaging consists of the following operations: filling of seed bags to an exact weight. So seeds has to be weighed properly and it has to be packaged in these package bags. Placing leaflets in the seed bags regarding improved cultivation practices. So why this seed is different? Why this seed is different than already existing seed? And for what purpose this seed variety has been invented? That has to be labeled. Attaching labels, certification tags on the seed bags and sealing of the bags. So labels and certification tags also includes certification tags, as I mentioned earlier. They come in different colors. So which will be easy for identification. Even the farmers or illiterates can easily understand. So these labels contains usually the purity level, the quality criteria, what are the tests, and for what purpose this seed variety is produced. So all these will be included in these labels. Finally, storage or shipment of the seed. So storage, again, depending on the duration of storage, the material, packaging material will be selected. As you can see in this picture, there are aluminum packaging, tin packages, glass jar packaging, and also you can see the gun HTPD bag, jute bag, barrier bag. So all these are different types of bags. We'll be choosing depending on what percentage of moisture and aeration is necessary for the seed, and also it will be depending on the variety of the seeds as well. And then seed certification. So seed certification is again has to be done before packaging, because certification tags has to be attached to the bags. So this comes under 5 important phases. First phase is verification of seed source. So whether the seed source is new one, whether it is already existing variety or it has undergone some improvement which is permittable for certification. Inspection of seed crop in the field. So crop performance, whether it is giving good yield or whatever the breeder has claimed the seed to be. So like good quality, good yielding seed. So whatever he has claimed, that has to be cross-checked. So seed certification agents will be visiting the field and they will be assessing whether whatever the breeder has claimed has been fulfilled. Next is supervision at post-harvest stages, including processing and packaging. So until the post-harvest, plantation till harvesting, the seed certifier will be assessing, will be gathering, correcting the informations, whether we have given right information also. Seed sampling and analysis, where some sample of seeds are collected from the harvest and it is sent for analyzing the germination, viability, purity, and the disease freeness, everything will be assessed. And lastly, certificate will be granted with the proper certification tags, labels, and sealing. So this is the complete process of seed certification. Lastly, seed marketing. So seed marketing comprises the following steps. Demand forecast. First, we have to understand what is in demand in the market. If it is off season for certain crops, those crops will be stored until the season is achieved. And if it is not in demand, the demand is created by marketing or by creating, it will be wait until the marketing has enough demand for the seed releasing. Marketing structure. So how you are going to market the seed, how you are going to reach the farmers through marketing techniques and tactics. Arrangements of storage of seeds. So until it is marketed, where you are going to store the seed and what are the storage structures you are going to use for marketing. And you can see sales promotion. So sales promotional activities where advertising will be necessary. Seed has to be given a common local name so that it will easily reach the market. It will easily be understandable by local farmers, and also certain advertisement using the well-known professionals for advertising and the preparation of pamphlets, ads, and so on should be performed. And post-sales services. So once you sell the seeds, so you should ensure that if the seed do not perform as expected, or if the seed is not purchased by the markets again back from the farmers, then we also should ensure the post-sale services. Companies or the breeders should ensure that the seeds will be definitely be accepted by the consumers, or it will be safe for using the seed sample, so that even in different environment, in different geographical conditions, they perform better. And lastly, economics of seed production and seed pricing. So this will be very important in the viewpoint of farmers and consumers, because if the seed is having all the good qualities, but if the pricing is too high, nobody is going to purchase the seed, because even though the yield is increased, if it costing the same and nobody is purchasing the seed, then it will be wasted. So to ensure that the seed pricing should be optimum compared to other available seed, it should be as low as possible, minimal as possible. So these are some marketing strategies and techniques that needs to be followed. Marketing organizations, there are central marketing cell, which will be giving its authority for regional offices. Under regional offices, there are many authorities that will be taking part in the marketing. So firstly, sole distributors, which is a private organizations, dealers, again the private organizations, and there are different types of dealers like private, cooperative, agro sale services, as well as the companies. So or the sale depots will be taking the role of marketing of the seeds. And coming to the final summary of the topic, seed production, it starts from the place of production where these general seed technology techniques, production techniques takes place in the places like institutes, organizations, research lab, seed companies, and the plant breeding organizations. And the methods of production to develop a new variety where different breeding programs and methods will be applied. And multiplication, as I said, pure seed are multiplied through different stages. Seed processing or treatment involves drying, cleaning, grading, disinfection, and protection. Seed storage, depending on the duration and purpose, it will be selected, and using proper equipment and temperature, we can store the seed better. Packaging and handling will involve different packaging materials and how we preserve the seed as long as possible. Naming of variety, the name of area, breeder, committee, or the year of production will be included in the naming of a variety in general. Seed certification through field inspection as well as laboratory tests will be necessary for certifying the seed. Release of an improved variety after approval from ICAR, the variety will come to the market. Publicity and distribution or marketing will be depending on the demand and need. Various organizations will be taking part in this. These are the reference materials for the topic, which you can refer for more information on any of the discussed topic. And thank you for listening. If there are any doubts, you can reach for me for clarity.